Hello everyone, let's get started. Today we will have the first of two lectures about microbial metabolism. And the focus of today's lecture will be enzymes and energy. Let's begin with a definition of this word metabolism. This term actually is defined as the chemical processes within a cell that are required to maintain life. As you can see, it's a very broad definition. It's not specific at all to any one particular process. When we look at all of the metabolism, therefore, that goes on in a cell, we have to have a way to categorize different groups of metabolic processes. And the first way we do that is by determining whether we're talking about anabolic metabolism or catabolic metabolism. So when we talk about metabolic reactions, again, chemical processes within a cell that are required to maintain the life of that cell, any reaction that involves the building of new molecules is an anabolic reaction. With regards to energy, an anabolic reaction is also considered endergonic. If you translate this term, it means consuming energy or requiring energy. Now compare that to all of the different types of metabolic reactions where molecules are actually broken apart. Those types of reactions are catabolic, and catabolic reactions are exergonic, which means they release energy. Now, as we've said several times in the course so far, one of the things that makes microbes so fascinating is that they have an incredibly diverse array of metabolic abilities. If you look across all of the different types of prokaryotic cells, both bacteria and archaea, you will find a tremendous array of different metabolic reactions that take place in those cells. Now, one of the ways we can place all of those diverse microbes into categories is by considering two things. How do they get their carbon every day in order to build molecules? And how do they get their energy to do that building? In other words, we can categorize microbes based on their energy class. Let's take a look at some of these categories. On this slide, we're looking at a diagram of how we place microbes into different energy classes. You can see up at the top, the first division involves whether or not the microbe gets energy from chemical sources, from actual molecules, or from the light from the sun. If you are a microbial organism and you can gather energy from sunlight, that's a process that we call phototrophy. You then would be called a phototroph. In comparison, if you get energy by breaking apart chemicals, you are a chemotroph. You are practicing chemotrophy. Now, when it comes to the phototrophs, the phototrophic organisms are literally harvesting energy from light and then using that energy in order to get ATP from it. Now, interestingly, this ATP that's produced through phototrophy is going to be turned right back around and used to build carbohydrate. You've all probably heard of the term photosynthesis, and photosynthesis is the combination of metabolic processes that ultimately produce carbohydrate, often in the form of glucose. Plants produce their own food from the light of the sun. Now compare that to the chemotrophs over here on the left. There are actually two classes of chemotrophic microbes. One class is able to get energy from organic molecules. And remember, we define that word organic as meaning carbon containing. There's also another class of chemotrophic microbes who are able to get their energy from inorganic chemicals. Things like hydrogen gas or hydrogen sulfide or ammonium, things like that. Things that don't contain carbon. If a chemotrophic microbe harvests energy from organic chemicals, we can call them chemoorganotrophs. They're going to take the energy that they harvest from molecules like, say, glucose, and they're going to use that energy to build, again, ATP. In the other category, any microbe that is a chemotroph and obtains energy from inorganic chemicals falls into the category called chemolithotroph. And again, they're able to harvest energy from molecules that do not contain carbon and use that energy to build. Are the kinds of reactions that break apart molecules. Another way to say that is, these are the types of metabolic reactions where bonds, covalent bonds, are broken. And when those bonds are broken, the pair of electrons that make up those bonds are released. And remember, it's electrons that are the conduits of energy in a cell. So catabolic reactions release energy by breaking bonds. Anabolic reactions, on the other hand, consume energy or require energy because they will be the kinds of reactions that build covalent bonds. Most biological reactions, whether they're anabolic or catabolic, are going to require enzymes. Now, enzymes are simply catalytic proteins. And remember, that word catalytic means able to enhance a chemical reaction. It's going to make a chemical reaction occur more swiftly. It's going to increase the rate of product formation in a chemical reaction. All enzymes are proteins. It's just that they have this special ability of catalysis. Enzymes are going to increase the rate of these critical biological reactions that compose metabolism in living cells. Now, something else to note, when you look at the name of an enzyme, the name will often tell you that it is an enzyme because most enzymes end with ASE. So if you see a word like amylase or peptidase or catalase, you know that you're dealing with a protein that is an enzyme. All proteins have a similar basic structure. All proteins are composed of amino acid subunits that are joined together in long, long chains. And the individual amino acids are bound to each other with a special kind of covalent bond called a peptide bond. We heard the word peptide bond when we were talking about peptidoglycan in the cell wall of bacteria. Remember, the chains of NAG and NAM in peptidoglycan have these short chains of amino acids sort of jutting off of them to the side. And between two strands of peptidoglycan, the short chains of amino acids will be bound together with peptide bonds, creating those cross bridges that give peptidoglycan its tremendous strength. So all proteins contain these peptide bonds holding together these long chains of amino acids. The chains of amino acids don't stay as linear structures, however. Once they're built, those individual amino acids in the chain are going to start reacting with each other through the building of things like hydrogen bonds and van der Waals interactions. Remember, every amino acid has a particular functional group sticking off of it, a side group. And that side group gives each amino acid its own unique chemistry. There are polar amino acids and non-polar amino acids. There are amino acids that have charged side groups, either positive or negative. And all of that individual chemistry is going to cause those amino acids to either be attracted to each other or repelled by each other. And all of that chemistry, that biochemistry, is going to cause that chain of amino acids to fold. It's the folding of that very immature protein that gives it a very complex three-dimensional shape. Every protein, each and every protein has its own unique shape. And that's because every protein has its own unique sequence of amino acids. It is the shape that a protein assumes when it folds up that will determine its function. And enzymes are no different. An enzyme must have the correct three-dimensional shape in order to do the job that it's intended to do. Take a look at this diagram at the bottom of the slide. The artist is showing us on the left-hand side this long chain of what looks like blue beads. And these blue beads represent individual amino acids. We can't see it, but remember that off of each one of these blue beads there is a functional group sticking out. And that functional group has some kind of biochemistry associated with it. So those functional groups are going to start pulling other things towards them, building perhaps hydrogen bonds or van der Waals interactions. Other functional groups are going to be repelled by neighboring amino acids. That's going to cause this to start folding. The first types of folding that we see are two varieties, what we refer to as alpha helixes. Those are going to start forming. And beta pleated sheets. These are more planar, two-dimensional structures. Then all of the helices and the sheets are going to start interacting with each other, and you're going to end up with a very complex three-dimensional shape. Every protein has its own unique sequence of amino acids. Every protein has its own unique shape. Enzymes are just catalytic proteins, and so it makes sense that every enzyme is different. Every enzyme is going to perform its own specific reaction. Now that reaction is going to occur the way all chemical reactions occur, whether they're metabolic reactions or not. If you remember from chemistry, the molecules that go into a reaction are often referred to as substrate molecules, or you may have heard the word reactant molecules. Those two words mean the same thing. When you put a catalyst in, the substrate is going to interact with the catalyst. A change is going to occur. Old bonds are going to be broken down. New bonds are going to be formed, and that will create product. After the product is formed, it will be released. In other words, it will stop interacting with the catalyst, and the reaction will be over. Enzymes are the catalyst in these biological metabolic reactions. They will bind with the substrate molecules, and they bind at a very particular location that every enzyme has that we call the active site. The substrate binds to the active site, and then old bonds are broken down, new bonds are formed, the product is produced, and then the product is released. Now, one characteristic that enzymes have, in fact, that all catalysts have, is that they themselves are not changed by the chemical reaction. They are not used up, in other words, by the chemical reaction. They can be used over and over and over again. Let's follow this diagram down at the bottom of the slide and see how this general enzymatic reaction occurs. In the drawing, the sort of beige colored shape down here, this represents the enzyme, and this yellow structure here represents the substrate. So this is what's coming into the reaction. It's not always a single thing. Sometimes there are multiple substrates that are going to come in together to a reaction. Now, the substrate is going to interact with the enzyme at a particular location, and that's this pie-shaped wedge that you can see here. The substrate is going to fit perfectly into that active site. That's important because we need that perfect interaction to occur. The next thing that's going to happen is, as I said, old bonds will be broken, new bonds will be formed, and our substrate will be transformed into product. And in this drawing, the product is actually two separate molecules, the blue one and the green one. When the product is produced, the product no longer fits perfectly inside that active site, and so it gets released. Now the enzyme is ready to do it all over again. This new substrate will come in and bind to the active site. Old bonds will be broken, new bonds will be built, and product will be produced. That product is not going to fit very well into that active site, and so it gets released. And the enzyme is ready to do it all over again. On this slide, we can see another drawing of an enzyme and a substrate molecule. Now this drawing is a little bit more realistic. The enzyme is depicted by the green molecule here. And I want you to notice that this is actually made out of maybe hundreds or thousands of little green circles. Each one of those green circles represents an amino acid. Proteins tend to be enormous molecules, relatively speaking. Most biological proteins contain hundreds or even thousands of amino acids. So when those amino acids interact with each other and that protein folds up, you get all kinds of different shapes. And the shape is what determines the function of that protein. So this enzyme is folded in a way that creates this active site. And yes, many active sites are actually sort of divots or cupped, concave areas on the molecule, places for the substrate to come in and bind. Now this drawing represents an enzyme called hexokinase, which is an enzyme involved in the metabolism of glucose. And the substrate, which is drawn as this little red molecule, is glucose itself. Notice how small the glucose is relative to the hexokinase. That's realistic. So the glucose is going to come in and it's going to land and bind in that active site. When it comes in, the molecule of glucose is going to interact with the amino acids that are around it. Remember your biochemistry. There will be some polar interactions and some nonpolar interactions. There may be interactions involving positive charges and negative charges. When two molecules come together, they will interact with each other based upon their chemistry. So what the artist is showing us is that when the glucose comes in and binds at the active site in this enzyme, the whole thing is going to change shape. Can you see that? That divoted area, that sort of pie-shaped wedge here, that is going to change because there's interactions going on in here. The whole thing becomes a new molecule. It's what we refer to as the enzyme substrate complex. And this change in shape is really critical because it's the change in shape that's going to allow for those old bonds to be broken and the new bonds to be built and product to be produced more quickly.